So, good evening to one and all present here. So, myself, Ajit Kumar V, a PhD research scholar from Department of Bioscience and Bioengineering from IIT Guwahati. So, this is going to be the last live interaction session, which is week 12 live interaction session for the course Environmental Biotechnology with the course code NOC 25BT83. And I hope my voice and PPT is visible.
Yes, sir.
Okay, then we will move on to the lecture. So before moving on to the lecture, I will briefly introduce about myself. Myself, Ajit Kumar, a joint degree research program scholar, which is a research collaboration between IIT Guwahati and IIT BHU Varanasi. I work in the laboratory of biochemical and environmental biotechnology, where our lab focuses on different wastewater treatment across different industries, and we use different techniques in order to handle them. And my research inclines on the application of enzymes, especially in the textile effluent. Moving on to my qualification, I have done BTech Biotechnology from Shastra University and MTech Industrial Biotechnology from NIT Surathkal, Karnataka. So moving on to my work experience, I have worked in many pharma companies like Biocon, Enzyme Bioscience, etc. This is all about my personal introduction. And in this week 12 live interaction session, as we have in the earlier weeks, we have already discussed all the important topics. So this session will be solely on discussing the MCQs. So as early lectures, I will be displaying the questions along with the options. You just take some time to read the questions and think about it, and you can put your answer either in the chat box or you can unmute your mic and you can answer it. So this week is solely on MCQ. And last class we have seen all the applications on the bioremediation, how it is applied in the real-time condition. We have also seen numerous case studies associated with it, and we also seen the heavy metal problem, how it is treated, etc. So these are all the concepts that has been covered, and for today's week 12, we will be seeing the MCQs one by one. So without any delay, we will move on to the first MCQ, which is biosurfactant primarily help in the degradation of option A, heavy metal, option B, insoluble nonpolar molecules, option C, soluble nonpolar molecules, option D, insoluble polar molecules, option E, soluble polar molecules. So among this, biosurfactant primarily helps in degrading which of these molecules? Option A, whether it is an heavy metal or insoluble nonpolar molecules or soluble nonpolar molecules or insoluble polar molecules or soluble polar molecules. Option D, okay, insoluble polar molecules. Okay. So any other answer? You just read the question carefully and we will be seeing the answer. biosurfactant primarily helps in degradation of which of this molecule? Okay, we will see the answer now. The option is the correct answer is option B, which is insoluble non-polar molecules. Why? We will see in the solution. So, first of all, the insoluble non-polar molecules can be solubilized and they are made available via biosurfactant. What the biosurfactant does? It will gather all the non-polar that are insoluble in nature. First, they will accumulate all the pollutant which are insoluble and non-polar. Then they will try to solubilize them using the biosurfactant. And they go move it for bioavailability so that it can be degraded by microorganism. So the correct answer is insoluble nonpolar molecules and the biosurfactant. Why it is very crucial for the biodegradation of these insoluble nonpolar molecules? Because they have the unique ability to attract both the hydrophobic and hydrophilic part. And we all know that hydrophobic means they are water hating. That is, they don't dissolve in the water. Whereas hydrophilic, they are water loving. That is, they easily dissolve in the water molecules. So this biosurfactant helps in bridging these both components in order to solubilize them. So what they essentially do, they encapsulate these insoluble nonpolar molecules, allowing them to expose to the water-based environments, which is aquatic environment. And thus they facilitate their degradation by providing appropriate microorganisms. So that is why the biosurfactant primarily help in degrading these insoluble nonpolar molecules. And we will see why other options are not relevant when compared to option B. So if we see the heavy metals, their primary function is not the degradation of heavy metals, right? So if we see the biosurfactant, this is not the primary goal. They what happens in that case? They helps to mobilize and expose these heavy metal that are trapped in the soil or other matrices, making more accessibility. So it is not the primary function of degrading the heavy metals. And if you see the soluble nonpolar molecules, as the option itself telling it is soluble, that is, they are already soluble, so it doesn't require the bridging function that the biosurfactant product provides. So here the usage of biosurfactant is not needed as they are already dissolved, right? So if you see the option D, insoluble polar molecules, as we know that it bridges hydrophobic and hydrophilic surface. So these molecules they are not going to enhance the interaction between already the polar substance that are present in the water. So this is also not appropriate of using the biosurfactant. And last option, it is same as the explanation of C. Since it isn't soluble, it is already dissolved. So it doesn't require any bridging function that it combines uh, hydrophobic and hydrophilic part. So the correct option is the biosurfactant is primarily used to degrade the insoluble or non-polar molecules. So we will move on to the next question. So the next question is, which of the following is the viable strategy to deal with toxic heavy metals? So among these strategies, which is most promising one to deal with these toxic heavy metals? Option A, complete oxidation. Option B, dissolution. Option C, precipitation. Option D, mineralization. Option E, dilution. So among these methods, which is found to be most viable when dealing with the toxic heavy metals? You can post your answers in the chat box or you can unmute your mic and you can say the answer. So among this, which is found to be most viable strategy to deal with heavy metals. So, which is the correct answer? Option C. Okay. Any other answer? Okay. Let's see the answer. Yes, it is correct. Option C, precipitation is the most viable strategy to deal with these kind of toxic heavy metals. And why it is so? Because precipitation is nothing but we are forming an insoluble compound that precipitate at the bottom of the tube, right? So if we are able to precipitate these heavy metals, what happens? The separation of heavy metals from the solution will be easier so that we can effectively trap these heavy metals and prevent them entering into the environment, right? So what happens? This precipitation reduces the bioavailability of these hazardous heavy metals. So among all the options, precipitation is the most viable strategy. And why not the other options? If we see the oxidation process, what if you remember the bioremediation of these heavy metals, what can we do with the heavy metals? We can only change the chemical state of the metals. That is, we can change, for example, in case of chromium, uh, we can we can change the uranium 6 plus to 4 plus, which is less toxic form. So it not necessarily remove the toxic metal by reducing or completely removing from the environment. So it doesn't oxidize and degrade them properly. So if you see the dissolution, obviously it is the opposite concept of the precipitation. So what happens? It dissolves the metals into the solution. If it dissolves, it will make more toxic by increasing the concentration of the heavy metal in the aquatic environment. So this is also not a viable strategy. Whereas in the option D, mineralization, as we have already know, metals cannot be completely mineralized, but we can reduce their concentration by moving on to the different strategies like precipitation. And if we see the dilution, what happens? Dilution means we will be adding more amount of aquatic solution, either water, in order to reduce their concentration. So what happens here? It doesn't actually remove the metals from the system. It is just decreasing the concentration of the heavy metal. But in case of precipitation, once we precipitated these toxic metals, thus it settles at the bottom of the tube. So this easier separation can be achieved when compared to all other options. That is why the precipitation is the most viable strategy to deal with the toxic heavy metal. So I hope you have understood the options and their solution. Then we will move on to the next question, which is the toxicity of the metal is mostly due to their affinity for the dash group of proteins. So, So the toxicity of the metals is mostly due to their affinity for which group of proteins? Option A, methyl. Option B, formyl. Option C, carbonyl. Option Option C, carbonyl. Option D, amine. Option E, sulfhydryl. So among this, which has the most affinity to interact the metals and the proteins? Carefully read the question and you can post your answers in the chat box. Toxicity of the metal is mostly due to their affinity for the which group of proteins, whether it is a methyl group or formyl group or carbonyl group or amine group or sulfhydryl group. Okay, option B. Option E. Okay, so any other answer? You can carefully read the question. You can carefully read the question and you can post your answers in the chat box. Okay, what is the answer? E. Okay, E. Then we will see the answer. Yes, it is correct. The correct answer is the E, sulfhydryl group. You both said the correct answer. We will see why the sulfhydryl group. Because if you see the sulfhydryl group, they are the highly reactive functional groups. And where it is found, it is mainly seen in the cysteine amino acid within the protein. And we all know that the protein is made up of amino acid. And if it's in amino acids, there are more than 20 essential amino acids, among which the cysteine is one of the amino acid that has SH groups, and these SH groups has the strong tendency to attach to the metal ions. Once this sulfhydryl group attaches to the metal ions, what happens? It will going to disrupt or destroy the protein structure and eventually affects its function, leading to the toxic effect. That's why sulfhydryl group is the correct answer. And what about the other options? If you see the methyl, they are generally non-reactive and they do not form a significant bonds with the metals, with the proteins. So these are non-reactive. And if you see the formyl groups, these are relatively unreactive when compared to sulfhydryl, and these are all not primary target for metal binding. And if you see the carbonyl group, uh, this carbonyl group may interact with the heavy metals, but they have weaker affinity than the sulfhydryl group. And same applies to the amine group. These are also the common groups that interact with the toxicity metals, but their affinity is very low when compared to the sulfhydryl group. That is why the correct answer is sulfhydryl. So I hope you have understood. So we will move on to the next question. So the next question is, which of these following process tends to yield high energy? So I will repeat the question. Which of the following process tends to yield higher energy? Option A, bioaccumulation. Option B, bioabsorption. Option C, bioreduction. Option D, biomineralization. Option E, bioleaching. Which of these? So which of the following process yield high energy? Bioaccumulation or bioabsorption or bioreduction or biomineralization or bioleaching? You can post your answers in the chat box. So what is the answer? Option Option C. Okay. Any other answer? I will repeat the question. Which of the following process tends to yield energy? Bioaccumulation, bioabsorption, bioreduction, biomineralization, and bioleaching. Any other answer? Option C. Okay. Any other? So we will see the answer now. Yes, it is correct. Option C, bioreduction is the correct answer. Why it is so? Because it tends to reduce the concentration of the pollutant by oxidizing them so that oxygen will be used as an electron acceptor and so it helps in degrading the substrate along with high energy. And what about the other options? If we see in case of bioaccumulation, there will be an uptake of metals or the substance and it will be degraded by microorganisms. So it is not going to generate any energy. Whereas with respect to the bioabsorption, as we have already studied in the earlier lecture, it helps in passive binding of the substrate to the surface of the microorganism. So it is just an binding physical process. So it doesn't generate any outcome of energies. Whereas in biomineralization, here what will be carried out formation of inorganic mineral compounds will be formed by the microorganism, so it is also not directly yielding the energy as compared to bioreduction. Whereas in bioleaching, it can generate the energy through oxidation but not solely about the energy generation. It needs certain other chemical substance in order to generate energy, whereas in bioreduction it can solely yield energy. So among these statements, bioreduction is the most appropriate process tends to yield the energy. So we will move on to the next question. Which of the following is regarded as simplest way to remove the toxic metals? Option A, bioaccumulation. Option B, biosorption. Option C, bioreduction. Option D, biomineralization. And option E, bioleaching. So, which is the simplest way to remove the toxic metals? Which is the simplest way? Whether it is in bioaccumulation or biosorption or bioreduction or biomineralization or bioleaching. You can post your answers. Which among the following process is the simplest way to remove the metals? Option B. Okay. So any other answer? You can read the question carefully. Which of the following is regarded as simplest way to remove the toxic metals? Your answers? You can post your answers. So which of the following is regarded as simplest way to remove the toxic metal? Option C. Okay. Any other answer? Okay, we will see the answer. Yes, it is correct. The option B, bioadsorption, is the correct answer. Why it is so? As we have seen previously, bioadsorption is nothing but it is a physical adsorption process. So what happens here? There will be live microorganism or the dead biomass will be utilized to absorb these heavy metals. How do they absorb? They absorb by attaching on the surface of the cell through various interaction that remove them from the surrounding environment. So here there is no energy involved. Here only the dead biomass or live biomass is required. So it is considered to be simple and it doesn't require any complex metabolic activity or transformation of the metals. It is just the metal and the biomass will be binding to it and it will be separated from the environment. So among all the options, the biosorption is the simplest way and more economical when compared to all other options. And why not the other options? If we see in the bioaccumulation, microorganism accumulate these toxic metals inside them. And this process is considered to be more complex as it involves various active transport. If there is any active transport, then only the heavy metals will be entering into the cells, and it is mainly influenced by the cellular metabolism. So there is a large complex active transport and cellular metabolism involved. That is why it is more complex when compared to biosorption. Whereas if we see the bioreduction, it encompasses the broad range of mechanism. It can include any way of bioremediation. Whereas if we see the biomineralization, so it is like a forming the solid metal precipitate by the microorganism so that it can immobilize and trap the toxic metal. So it is more complex due to the various biochemical reactions that are occurring in the system. Whereas if you see the bioleaching, what happens here, they will be using microorganism to dissolve and leach the metals from the solid materials. So here what happens, it is going to involve various complex enzymatic reactions and they are more intricate. That's why among the all the options, biosorption, it is like a physical surface phenomena where the metal ions will be binding onto the surface of the cell. Thereby, we are removing them from the environment. So it is more cost effective and simpler procedure when compared to all other options. Then we will see the next question. Biohydrometallurgy is most extensively used for which of the following metal recovery or mining? So, biohydrometallurgy is the most extensively used for which metal? For recovery or mining? Option A, copper. Option B, gold. Option C, uranium. Option D, iron. Option E, coal. So, from this, which metal is recovered by the process of biohydrometallurgy? You can post your answers. So, which is the metal that is used to extract through biohydrometallurgy? Is it copper or gold or uranium or iron or coal? You can post your answers. Option A, okay. Any other answer? Option A. Option A. Okay. So we will see the answer. Yes, it is correct. Option A. First, before moving on to the discussion, we have to understand what is biohydrometallurgy. Bio is nothing but usage of microorganism, that is biological source. And hydro means a process that occurs in the aquatic environment, that is water. And metallurgy related to process involving the separating and refining the metals from the raw ores. Now you could understand that. So it is simply a bio-mining or extracting the valuable metals from the low-grade ores. How do you extract? Using some microorganism. Mostly Ferredoxin species are involved in extracting these species metals from low-grade ores. And if you see among the various method, copper is the metal that are most recovered using the biohydrometallurgy. Why it is so? Due to the presence of sulfide minerals, chalcopyrite, which can be oxidized by the bacteria to solubilize the copper. And if you see, it has been estimated that across globally 10 to 15 percentage of the copper production are extracted through this biohydrometallurgy, whereas only 5 percentage is only extract is used for extracting the gold, and they are depend on the biohydrometallurgy. So almost 15 percentage of the copper they have been extracted using the biohydrometallurgy. And why not the other option? As we have seen, the gold, it is also used, the biohydrometallurgy is also used for gold recovery, but they often involves other chemicals like cyanide, leaching. So, and we, from the fact also, we have observed that only 5 percentage of gold is recovered through biohydrometallurgy, whereas through copper, almost 15 percentage of the copper is extracted through this technique. And if you see the uranium, um, so this technique is not as prevalent as the copper. It has potential for uranium recovery, but not directly involved. Whereas in iron, this is not primarily not to extract the metal itself, but rather to create the oxidizing agent. So they are not using for directly extracting the ions. So it was used to create an oxidizing agent like ferric sulfate, which helps in leaching the other metals like copper. So iron is not directly involved. Whereas the coal, as we already know, coal is a fossil fuel. They are not metal, right? So it is not recovered through biohydrometallurgy. So among these options, the widely used is copper recovery or mining. We will move on to the next question. Which of the following elements can be used as an electron carrier during the microbe mediated indirect metal oxidation? So which among these element used as an electron carrier during the microbe mediated indirect metal oxidation? Whether it is a option A hydrogen, option B nitrogen, option C carbon, option D iron, option E sulfur. So among this, which element is used as an electron carrier during microbe mediated indirect metal oxidation? Which acts as an electron carrier? Option D. Okay. Any other answer? Option D. Okay. Now we will see the answer. Yes, it is correct. Option D. Why it is? Because iron can exist in both forms, Fe2 plus and Fe3 plus oxidation state, right? So it allows readily to accept or donate electrons. So this makes it as a suitable electron carrier during indirect metal oxidation. And what is indirect metal oxidation? The microbes do not directly involve in the metals to extract them. So first the microbes will oxidize this Fe2 plus to Fe3 plus, and this Fe3 plus helps in oxidizing this metal sulfide, converting into metal ions. So the metal sulfide plus Fe2SO4 thrice, what will it form? First it will form ferric sulfide followed by the metal sulfide, so that it precipitates. The metals can be precipitated through indirect metal oxidation approach through microorganism. So this is why the iron can be used, because it coexists in both the oxidation state, which is 2 plus and 3 plus. What about the other options? If we see the hydrogen, it acts as an electron donor in certain metabolic pathway, right? Not in the metal oxidation pathway. That's why it is not the correct answer. And if you see the nitrogen, as we have already seen, these nitrogens they play important role in the nitrogen fixation and other nitrogenous related process, and they are not involved in any metal oxidation process and acts as an electron carrier. Whereas carbon, and we know that these are all the backbone of the organic molecules and helps in synthesizing the biomass and energy production. So they are not involved directly in the electron transfer as in case of metal oxidation. If we see the sulfur, sulfurs what they do normally they involve in the redox reaction. So they involve in the sulfur metabolism, for example. So this is not directly involved in the metal oxidation. So that's why among the statement, iron is used as an electron carrier during the microbe mediated indirect metal oxidation, where metal is precipitated by using the ferric sulfide. So we will move on to the next question. Which one of the following options presents the correct sequence of event leading to the eutrophication? Option A, organic matter decomposition, oxygen depletion, nitrite production, and phosphate solubilization. And option B, organic matter decomposition, phosphate solubilization, oxygen depletion, and nitrite production. Option C, phosphate solubilization, oxygen depletion, nitrate production, and organic matter decomposition. Option D, phosphate solubilization, nitrite production, oxygen depletion, and organic matter decomposition. Whereas option E, organic matter decomposition followed by phosphate solubilization followed by nitrite production, then oxygen depletion. So among these, which is the correct option that presents the correct sequence of event leading to the eutrophication? You can post your answers. So which one of these following options present the correct sequence of event leading to eutrophication? Option D and option E. Okay. Any other answer? Answer option A. Okay. So now we will see the answer. Correct answer is option A. Organic matter decomposition, oxygen depletion, nitrite production, phosphate solubilization. Yes, the correct answer is option A. We will see the explanation. First, what happens? The organic matter decomposition occurs. So, when these dead organism decomposes, what happens? The bacteria comes into action and break down these organic matters, which releases the nutrients like phosphorus and nitrogen into the water. So the decomposition of organic matter occurs and they release phosphorus and nitrogen. Then the bacteria will solubilize these phosphorus, making it available to uptake by the algae. Then the nitrogen is still there. So with the increased availability of nitrogen from the organic matter that we have already seen, the bacteria will convert these nitrogen into nitrite through the process of nitrification. As the bacteria utilizes all the phosphorus and nitrogen, they consume oxygen, leading to the depletion of dissolved oxygen in the water body. This leads to the depletion of dissolved oxygen, which leads to eutrophication. So, this is the overview of how eutrophication is done. First, the organic matter will be decomposed by the bacteria into nitrogen and phosphorus and many other organic matter, and these phosphorus will be solubilized. Then the nitrogen will be converting to nitrite, so that nitrogen and phosphorus will be fully utilized by these microbes and algae. Once it got depleted, then they will go for the oxygen and causes dissolved oxygen depletion in the water bodies, finally leading to eutrophication, which affects the complete aquatic ecosystem. That is why the option A, first the organic matter decomposition will be taking place, then followed by the other options. Then the next question is, which of the following is not a major member of EBPR microbial community? Option A, denitrifiers. Option B, microthrix. Option C, nitrosomonas. Option D, streptococcus. Option E, accumulate bacteria. So, which of these following is not a member of EBPR microbial community? And anyone knows what is EBR? EBPR? Anyone know EBPR? Okay, answer is D, Streptococcus. So what is EBPR? Anyone? What is the expansion of EBPR? It is an enhanced biological phosphate removal. So which is the not a major member of this microbial community. Yes, it is enhanced biological phosphate removal, correct? Option D. Okay. And we will see the answer. The answer is option C, which is nitrosomonas. So this is not the member of the EBPR, which is enhanced biological phosphorus removal. Why it is so? Because these nitrosomonas, they are commonly involved in the other activity, which is nitrification. And what is nitrification process? Converting the ammonia to nitrite rather than the phosphorus removal. That's why the nitrosomonas is not considered as a major member of the EBPR microbial community. The next question, the conversion of ammonia to nitrate is known as, I hope you have read the lectures on nitrogen metabolism, where different terminologies we have studied, nitrification, denitrification, etc. So, what is the process called conversion of ammonia to nitrate? Option A, denitrification. Option B, nitrification. Option C, nitration. Option D, nitration. Option E, nitrification. These are all seems to be confusing. You just concentrate on the terms, that is what is we call conversion of ammonia to nitrate. Option E, okay, nitrification. Any other answer? Okay, so what is the answer? What do we call ammonia to nitrate? Option E. Okay, let's see the answer. Yes, it is correct. Nitrification. So we all know that nitrification is a two-step process where first bacteria will be converting ammonia to nitrite, then nitrite will be converting into nitrate. So we call this whole process as nitrification, right? So that's why the correct option is nitrification. So what is nitriation means? So it is a different process where oxidation of nitrogen in ammonia to nitrate. And nitrification. What is mean by denitrification? It is just opposite to of what we have seen, the nitrification. So what happens? The nitrate will be converting into ammonia, right? Nitriation means it will be adding atmospheric nitrogen, that is nitro group, to the molecule. So it does not involve in any nitrogen metabolism. This is a chemical reaction where NO2 group will be added to the molecule. So these are all different terms, but the actual conversion of ammonia to nitrate indicates the nitrification process where first step will be ammonia to nitrate, then followed by nitrate. So we'll move on to the next question. Yes, it is correct. The both these steps, that is ammonia to nitrite to nitrate. So the next question is the overall process of nitrogen removal involves the conversion of which compound to which compound. Here we are talking about the overall process. Overall process of nitrogen removal involves the conversion of option A ammonia to nitrite, option B ammonia to nitrate, option C ammonia to nitrogen, or it is nitrogen to ammonia, option E nitrogen to nitrate. So among this, which describes the overall process of nitrogen removal. And your answers are option C. Okay. Ammonia to nitrogen. Okay, we will see the answer now. Yes, it is correct. It describes the overall process, ammonia to nitrogen. What happens? The ammonia and their oxides of nitrogen, they normally present in the wastewater. And these acts as a pollutant, that is source of pollutant for the microorganism. And they help us to convert into inert nitrogen gas. So the correct option is ammonia to nitrogen. And what about the other options? What do we call ammonia to nitrite conversion? So this is a part of nitrification process, right? The first step of nitrification process. So it doesn't describes the overall process of nitrogen removal. And what is the overall process of nitrogen removal? First, the ammonia will be converting into nitrite, then it will be converting into nitrate. We call this process as nitrification. Then again the nitrate will be converting into NO2. thus completing the nitrogen cycle. Then nitrogen fixation will be happening where the atmospheric nitrogen will be converting into ammonia. And if you see the ammonia to nitrate, we call this whole process nitrification. Yes, it is correct. And this is the one step in nitrogen removal, not the entire cycle. Whereas if you see nitrogen to ammonia, so this is actually the reverse of the nitrogen fixation. So this is also a wrong answer. Whereas nitrogen to nitrate, so this is also not a complete removal process, right? So these are all a part of nitrogen removal process, and the only option which is ammonia to nitrogen provide us the whole process of nitrogen removal. So the next question is the ammonia oxidizing bacteria are preferred in the modern biological nitrogen removal system. So which of these following is false about the system? Option A, they require less organic carbon. Option B, they require less oxygen. Option C, they produce less sludge. Option D, they produce more nitrate. Option E, they require inhibition of nitrite oxidizing bacteria. So the question is the ammonia oxidizing bacteria, they are preferred in modern biological nitrogen removal system. Among these statement which is false about the system, whether it requires less organic carbon or it requires less oxygen or they produce less sludge or they produce more nitrate or they require inhibition of nitrite oxidizing bacteria. Option B, okay, they require less oxygen. Any other answer? Option B, okay. Let's see the answer now. The correct answer is they produce more nitrate. So this is the false statement about the system, and we will see why it is so. Because if we see the ammonia oxidizing bacteria, they will be skipping the process of oxidation of nitrite to nitrate. So since they are skipping this oxidation process of nitrite to nitrate, so there will be no more amount of nitrate will be produced during the system. That is why this is false. And if you see the other options, these ammonia oxidizing bacteria, they are normally chemoautotrophs. That means they get the energy from the inorganic compounds like ammonia, not the organic compound. So this is correct because they require less amount of organic carbons when compared to the inorganic. And if you see the next option, they require less oxygen. Yes, while they do so, while they do use the oxygen to oxidize this ammonia compared to other biological process. And also converting the ammonia to the nitrite, it requires less oxygen than the converting to nitrate. So this leads to the energy saving in the aeration system. That is why, yes, they require less oxygen so that energy will be saved during the process of aeration. So this is also correct. And if we see the third option, yes, it also produces less sludge production compared to the conventional treatment of nitrogen removal. And the last option, for example, for an efficient nitrogen nitrate shut process, it is crucial to inhibit these nitrite oxidizing bacteria to prevent what? To conversion of nitrite to nitrate. So the option A, B, C, E are correct, whereas option D is wrong about the system since these ammonia oxidizing bacteria is already going to skip the for oxidation of nitrite to nitrate. So the overall nitrate concentration will not be present at the very high concentration. So we will see the next question. N-Damo archaea offers an unique advantage in the biological nitrogen removal by virtue of their ability to use which as an electron donor in terms of N-Damo archaea. Option A, ammonia. Option B, nitrite. Option C, hydrogen. Option D, methane. Option E, methanol. So, the N-Domoarchaea offers an unique advantage in biological nitrogen removal by virtue of their ability to use which one of these as an electron donor. You can post your answers here. You can read the question also. Option A, okay, ammonia. Any other option? Option D, methane. Okay, we will see the answer. Yes, it is correct. The option D is the correct answer, methane. Before that, we have to understand what is N-Domoarchaea. It is nothing but NO3 dependent anaerobic methane oxidizing archaea. So what happens? This type of archaea, they will be using methane as the source of energy to reduce nitrate or nitrite to the nitrogen gas. So this is one of the crucial process for nitrogen removal in the wastewater treatment. So first, if you understand the term, which is NDAMO, which is nothing but NO3 dependent anaerobic methane oxidizing archaea. So it will be using methane as one of the source. What happens here? They will reduce the NO3 to NO2 with methane as an electron donor, which offers a more reliable nitrite supply to the ammoniacs than the ammonia oxidizing bacteria, which eliminates the requirement of nitrogen oxidizing bacteria suppression. That's why here methane will be utilized as an electron donor. And what about the other options? If you see the ammonia, yes, it acts as a nitrogen source for other microorganisms like Ammonox bacteria, but not to the Endomoarchaea. And option B, nitrite. What is nitrite? We have already seen it is one of the intermediary product in the nitrogen removal process. So definitely it is not going to act as an electron donor for Endomoarchaea. And if we see the option C, hydrogen, some microorganism may utilize the hydrogen as an electron donor, but Endamo they have adapted to utilize methane as an electron donor than the other substrates. And methanol, This is also, it is generally a not an electron donor as we have studied in the previous lectures, and especially with the nitrogen removal, uh, we aren't aware that methanol has been utilized as the electron donor, right? So the correct answer is methane. So we will move on to the next question. Which of the following microbial product is correctly matched with its application? So here the left hand side is their microbial products, right hand side it is the application for in oil recovery. So which is the correctly matched? Option A, biomass for oil dissolution application. Option B, biopolymers for rock wettability alteration. Option C, solvents for viscosity reduction. Option D, acids for oil degradation. And option E, gases for oil emulsification. So among this, which is correctly matched with oil recovery application? You can read the question carefully and you can answer. Option C and option B. Okay. Option B, biopolymer. Option C, solvents. So let's see the answer now. The correct answer is option C, which is solvents for viscosity reduction. And why it is so? The solvents that are produced by these certain microbes, they directly interact with the oils. For what purpose? For reducing their viscosity and making it easier to flow through the porous rock formation during the extraction process. That's why the solvents that are produced by the microbes, they help in reducing the viscosity so that it can easily flow through the porous rock, these solvents, and it helps in extraction process. And what about the other options? If you see the biomass, and we already know biomass is a microbial product and they are not involved in the oil dissolution, right? Because it will be a substrate for the microorganism. And if you see the biopolymers, they are more specifically used for mobility control and selective plugging. So their application in rock wettability alteration is not found. And if you see the acids, they are known to dissolve the carbonate rocks, not directly involved in the oil degradation. Whereas in case of gases, they contribute to the reservoir repressurization, oil swelling, and viscosity reduction, not in the oil emulsification process. That's why the correctly matched is the usage of solvents in the viscosity reduction, especially in the degradation recovery of oil resources. So the next question is the most suitable genus for microbially enhanced oil recovery is. Among this genus is mostly widely used. Option A, Klebsiella. Option B, Clostridium. Option C, Pseudomonas. Option D, Heterobacter. Option E, Methinobacterium. So among this, which suits best for microbially enhanced oil recovery? option E and option A. Okay, option E, option A. Okay, we'll see the answer now. The correct answer is option B, the Clostridium. Why? Because they have the ability to produce various metabolites that are beneficial for the oil recovery. What kind of metabolite? Solvents like butanol and acetone is generated from the Clostridium species, and gases like CH4 and carbon dioxide and organic acids too. And additionally, we also know that Clostridium species is obligate anaerobes. What do you mean by obligate anaerobes? They are strictly anaerobic in condition. That is, they do not want oxygen to be present in their environment. So, this is one of the most advantageous for many oil reservoirs because we have seen many case studies where in oil reservoirs there is an anaerobic condition most prevalent when compared to the aerobic. So, the Clostridium species is the suitable genus for microbially enhanced oil recovery since it has various metabolites that are beneficial for oil recovery. For example, solvents, gases, and organic acid. And if we see the other option, if we see the Klebsiella, they are used for solvent production, but Clostridium is more suitably due to its robust and multiple metabolism or metabolites that are generated for particularly enhancing the oil recovery. And if you see the Pseudomonas, it is generally used for biosurfactant production and helps in degrading the hydrocarbons that are present in the microaerophilic or aerophilic conditions. Whereas Heterobacter, they are used in the microbial enhanced oil recovery also, but not at as convenient as the Clostridium because there it produces various metabolites like solvents, gases, and other metabolites. If you see the methanobacterium, yes, it is used for normally for methane production, which contributes for the reservoir reprecision and oil spilling, not for oil recovery. So that's why Clostridium is more appropriate species when compared to the other options. The next question is which of the following options is most appropriate to state the key difference between the bio-based plastics and biosynthesized plastics? So what is the key difference between these two? Option A, bio-based plastics are biodegradable. Option B, bio-based plastics, they are made from biological raw material. Option C, biosynthesized plastics are biodegradable. Option D, biosynthesized plastics are polymerized by microorganism. Option E, biosynthesized plastics, they are made from the plant product. So among these, which best suits for knowing the key differences between the bio-based plastics and biosynthesized plastics? And your answers are option D. Okay. Any other answer? And option D. Okay. Okay. Option D, bio-based plastics are polymerized by microorganism. The correct answer is yes, it is correct. The option D, biosynthesized plastics are polymerized by the microorganism. So first, before moving on to the detailed solution, you have to understand the differences between the bio-based plastics and biosynthesized plastics. Bio-based are nothing but these plastics, they are made from the renewable biological sources like cornstarch or sugarcane. But these bio-based plastics may not be biodegradable, right? Because the plastics that are derived from the biological source, we call it as bio-based plastics. Whereas biosynthesized plastics, they are clearly identified by usage of microorganism to synthesis the polymer materials through polymerization process, where they synthesis the plastic material within their cells. So biosynthesized plastics, they are polymerized by the microorganism, while the bio-based plastics are polymerized artificially. So this is the key difference between the bio-based and biosynthesized plastics. And what about the other statements? Bio-based plastics are biodegradable. As we have seen, while some bio-based plastics can be biodegradable, but not all of them. So it is a separate characteristics from being the bio-based plastics. So this is wrong. Whereas in option B, bio-based plastics, they are made from biological raw material. It is true, but the statement is true, but it is not telling the key differences between the bio-based plastics and biosynthesis plastics. Whereas option C, biosynthesis plastics are biodegradable. This applies to the option A as we have explained, while some might be biodegradable, but this doesn't define the fundamental differences between these two plastics. Whereas option E, biosynthesis plastics, they are made from the plant source. While some can be derived from the plant source, but it doesn't define the characteristics difference between them. So the correct option that describes the key difference between these two plastics are biosynthesis plastics are polymerized by the microorganism. Which of the following statements about emerging pollutant removal system is false? So among these statement which is false regarding the emerging pollutant removal system. Option A, activated sludge systems, they are slow and inefficient. Option B, sorption process are used conjunction with the biological process. Option C, advanced oxidation process may use UV radiation. Option D, reverse osmosis systems, they are not effective. Option E, nitrification is used to degrade the estrogens. So among these statements, which is false? So which of the following about the emerging pollutant is false? Option F. There are only five options, Ashwini. Option D, okay to in. Option D, reverse osmosis systems are not effective. So any other answer? So let's see the answer. Yes, it is correct. Option D, reverse osmosis systems, they are not effective. Why? Why this statement is false? Because the RO reverse system, they are successful in removing the pollutant under the membrane-based technology. So these are all effective for cleaning these emerging pollutant. So this is the false answer. And what about the other options? Why they are true? And we all know, although these activated sludge systems, they are considered as an biological process, they can be very slow and inefficient, especially when dealing with the emerging pollutant. Since they are newer and persistent, they take long period of time to adapt and degrade these kind of pollutant. So yes, it is true. Whereas in the option B, sorption process are used in conjunction with the biological process. Yes, the absorption process means it's a biosorption, where we'll be using live or dead biomass for degrading the pollutant. But as we are focusing on emerging pollutant, it has to be combined with any other biological process to enhance the removal of emerging pollutant. Option C, advanced oxidation process may use the UV radiation. Yes, it is true. It frequently uses UV in combination with other oxidant like ozone or H2O2 process. Whereas option E, nitrification. So this is also one of the biological process. What happens here? Ammonia will be converting into nitrite, then nitrate. So it can contribute to overall removal of nitrogenous compounds, which indirectly impact the fate and behavior of certain emerging pollutant. So among these statements, reverse osmosis systems, they are not effective for emerging pollutant. This is the false statement. So moving on to the next question, the algae or autotrophic microorganism that can naturally convert CO2 to value added compound. Then why is there need of bioengineering these heterotrophic microorganisms for carbon capture and utilization? So as we already know, algae has also the capacity to convert the CO2 into value added compounds. But why we are going for and bioengineering of heterotrophic microorganism for carbon capture and utilization? Option A, algae are difficult to grow. Option B, algae have low efficiency. Option C, algae require expensive media component. Option D, algae produce harmful byproducts. Option E, algae requires precisely controlled temperature. Okay, Tugin. Key FS just beside. Okay, okay. Okay, option D. Okay, Ashwini. So what about the answer for this question? Option B, algae has low efficiency. Okay, what about any other answer? The question is very clear. We know that algae has also the capacity to convert the CO2 into value added compounds, right? So why scientists are going for bioengineering the heterotrophic microorganism for the carbon capture and utilization? Option B. Okay, Ashwini. So let's see the answer. Yes, it is correct. Algae has the low efficiency. So if you see the what is the low efficiency, the highest rate of lipid production by any algae is about 5 grams per meter square per day. But in order to commercialize them, and if we see the feasibility, at least we have to produce 30 grams per meter square per day. So that's why we are going for a bioengineered heterotrophic microorganism, since the threshold limit is 5 grams per meter square per day. What about the other options? Algae are difficult to grow. Mm, While the algae can be more challenging to grow on the larger scale than any other microorganism, but they are easy to cultivate in a controlled manner. So this is not the primary reason for going for an bioengineered heterotrophic microorganism. And also, if you see, the algae requires expensive media component. This might be partially true because some microalgae may require a specialized and expensive nutrient, but there are also other algae which can readily consume the easily available substrates and cost-effective substrates too. However, with proper strain selection and optimization of growth condition, harmful media component we can be overcome this expensive media component. Whereas if we see the algae produce harmful byproduct by proper optimization and media component condition, the harmful product can also be minimized and it can be managed. So you need to have a proper managing optimizing strategy. Algae require precisely controlled temperature. Yes, it does. They do require an optimal temperature condition for their growth, but this is manageable in a controlled condition. Temperature control is challenging, but it is not the primary reason. The primary reason for going for the bioengineered heterotrophic microorganism is that they often yield the low efficiency. That's why we are going for other mode of microorganism. Moving on to the next question. Plant-based carbon capture and sequestration aims to improve the efficiency by introducing the triad. So if we move on to the plant-based carbon capture systems, now they are using the plant also, but how they improve their efficiency, which triad they are introducing? Option A, taller shoots. Option B, shorter roots. Option C, longer lifespan. Option D, slow decomposition. Option E, fast decomposition. So among these triads, which one they are adapting for plant-based carbon capture and sequestration? Option D, okay, which is slow decomposition. We will see the answer now. Yes, it is correct. Slow decomposition. Why? Because in plant-based carbon capture sequestration, what is our aim? We will be capturing these carbons and will be converting into some useful product, right? Using the plant as the source. So by introducing the triad of slow decomposition, what happens? This allows the plant to store the carbon for longer period of time before breaking down, thus increasing the amount of carbon that is captured and stored in the ecosystem. That's why we'll be introducing the triad, which is slow decomposition. And what about the other options? Option A, taller shoots. If you see, while taller plant might have or might provide a higher surface area for capturing the carbon dioxide, but their height doesn't guarantee long-term carbon capture, where decomposition providing long-term carbon storage. If you see the shorter roots, what happens? If there is a shorter root, that might lead to less carbon uptake from the soil. So it not necessarily enhance the carbon sequestration in the plant itself. And option C, longer lifespan. Although longer lifespan contribute to increased carbon sequestration facility, by allowing the plants to accumulate more amount of carbon over the time, but the slow decomposition is the major factor which not only stores the carbon but also helps in converting for long period of time. Whereas option E, fast decomposition, what happens if there is a fast decomposition? It leads to the rapid release of the stored carbon back to the atmosphere. So defeating the purpose of carbon sequestration. So definitely option C is wrong. But among the all other options, option D, slow decomposition is most suitable trial that we will be introducing them in the plant-based carbon capture and sequestration. So we will move on to the next question, which is what is the correct order of the steps that are involved in converting a biomass into bioenergy? Option A, fermentation followed by hydrolysis stabilization. Option B, stabilization followed by fermentation and hydrolysis. Option C, stabilization followed by hydrolysis followed by fermentation. Option D, hydrolysis, stabilization and fermentation. Option E, hydrolysis, fermentation and stabilization. So which among the following option provides a correct order of steps involved in the conversion of biomass to bioenergy? Option E, okay. Any other option? Hydrolysis followed by fermentation, then stabilization. Okay. Any other answer? Option D, okay. Hydrolysis followed by stabilization, then fermentation. Okay. We will see the solution now. Option E is the correct answer, where hydrolysis greater than fermentation greater than stabilization. Why it is so? First, you need to understand the terms. Hydrolysis means this is the initial step where a complex carbohydrate or any other biomolecules that are present in the biomass will be broken down into simpler sugar. So, complex sugars will be converting into simple sugars like glucose by the action of numerous enzymes that are involved. So, once it got broken down, the next step is the biomass is often stabilized to prevent further degradation or decomposition. How it will be achieved? By adjusting the pH or adding these preservatives in order to stabilize these biomass for further degradation or decomposition. Then finally, the sugar that is released, that is these glucose that are released during the hydrolysis, they will be fermented by the microorganism in order to produce the biofuels or any other bio-valuable products like ethanol or methane. So that's why hydrolysis, then stabilizing the biomass because they should not degrade, then followed by the fermentation. So what happens if we see other options? Yes, the fermentation of the sugar requires what? First, the hydrolysis needs to be happen. Then only the fermentation will be carried out. So definitely option A will be eliminated. Whereas in option B, stabilization is generally done towards the end to preserve the products, right? So what happens? The sequence first hydrolysis needs to be there. So the option B and C will be eliminated. And if you see the option D, the fermentation comes after the hydrolysis to convert the hydrolyzed sugar to biofuels. That's why the option E is correct. First, hydrolysis will be happening. The hydrolyzed product will be converting into valuable bioenergy. Then the obtained bioenergy will be preserved for stabilization. So the next question is, Oh, okay. So in here, it is not the order of steps here. Here, we are trying to explain the terminologies. First, we will try the terminology for hydrolysis is the breaking down of complex substance into simpler product. So this is not the order of steps. This is for only definition. But the correct answer is yes, it is correct. Hydrolysis followed by fermentation followed by stabilization. Okay, this is for explaining the terminology of what it is. The correct answer is C. So the next question is biohydrogen cannot be produced via which of this mechanism? Biohydrogen. Option A, photofermentation. Option B, anoxygenic photosynthesis. Option C, water splitting photosynthesis. Option D, microbial electrolysis process. Option D, dark fermentation. So, which of these following methods by which the biohydrogen cannot be produced? Option B, okay, anoxygenic photosynthesis. Any other answer? Option C, okay, water splitting photosynthesis. So, we will see the answer. The correct answer is anoxygenic photosynthesis. Why it is so? So what happens? It typically utilizes hydrogen as the electron donor and oxidize them into water. So in this process, what happens? They will be utilizing the hydrogen. They do not produce the hydrogen. In anoxygenic photosynthesis, they will utilize the hydrogen and they make water, not the reverse process. So, among the other statement, an oxygenic photosynthesis is the wrong answer, which cannot produce the biohydrogen. And if we see the photofermentation, yes, by photofermentation we can generate biohydrogen. What it is? It is a process they use photon, which is the light, in order to produce the hydrogen through fermentation of organic substrate by certain microorganisms. So this photofermentation will help in producing the hydrogen with help of the light. And if we see water splitting photosynthesis, it is nothing but if you see a certain microorganism like cyanobacteria, they can produce hydrogen through photosynthesis. But they use this hydrogen internally by their own metabolic process, not directly. And this is also one of the process where water will be splitting into hydrogen and oxygen. So eventually through photosynthesis process, H2O will be converting into H2 plus O2. So this is also one of the process where hydrogen is produced by certain microorganism. Whereas option D, microbial electrolysis process, what happens? They used to generate the electricity to drive the microbial metabolism, producing hydrogen as the resultant product. And if you see the option E, the dark fermentation, where we will be using anaerobic bacteria to produce the hydrogen without the need of any light. So the option A and option E, these are all two different fermentation where hydrogen is produced. And the correct answer which cannot be produced through biohydrogen is the anoxygenic photosynthesis. Why it is so? Because here we will be using the hydrogen to produce water. So the next question is at Hartford, USA, the chromium contamination was treated using the polylactate amendment. So here the polylactate acts as a option A, it became the electron donor for chromium 6 reduction. Option B, it became electron acceptor for chromium 3 oxidation. Option C, it became carbon and electron source for iron reducing bacteria. Option D, it became carbon and electron source for ion oxidizing bacteria. Option E, became carbon source and electron sink for ion reducing bacteria. So among this, which is the correct answer? Option C. Okay, we will see the answer now. Yes, it is correct. Option C. Polylactate became the carbon as well as the electron source for ion of reducing bacteria. Why it is so? The bacteria need one carbon source and electrons. They need carbon and electron for their growth and energy, right? So in this case, these polylactate will be acting as an both carbon and electron source, and this will be this polylactate, which is a biodegradable polymer of lactic acid. So this will be broken down by the appropriate microorganism. What kind of microorganism? It is an ion reducing bacteria. So these bacteria will be utilizing the ion 3 as an electron acceptor in their respiration, reducing it to ion 2. By providing a carbon source like polylactate, these bacteria will be actively reducing the ion 3 while simultaneously obtaining the energy for their own metabolism. So that's why polylactate, it acts as both carbon and electron source, especially for ion reducing bacteria. What about the other options? ABC became polylactate became electron donor for chromium 6 reduction. While chromium 6 reduction is important, but polylactate, they are not directly involved in the electron donation to the chromium 6. So this is not suitable for the case study that we have been seen in the chromium contamination. If we see the second option, the chromium 3 oxidation, which is nothing but converting chromium 3 to chromium 6, which is more toxic, right? So this polylactate will not be act as an electron acceptor and does not participate in any oxidizing process. And if we see in the ion oxidizing bacteria, what happens? The electron acceptor is oxygen, not the ion 3. While the polylactate will be acting as a carbon source, it will be unlikely utilized by these kind of bacteria due to their specific oxygen requirement. And if we see option E, although polylactate acts as a carbon source, but it doesn't act as an electron sink for ion reducing bacteria. It It in this case, what happens? The Fe3 is acts as an electron acceptor, not the polylactate. So the correct answer is the polylactate will be acting both as an carbon and electron source, particularly for ion reducing bacteria, not the ion oxidizing bacteria. So the next question is, under certain circumstance, these ion reducing bacteria can reduce the uranium. This is possible because option A, iron and uranium have similar reduction potential. Option B, iron and uranium have similar mass. Option C, iron and uranium have similar structure. Option D, iron and uranium have similar charges. Option E, iron and uranium have similar size. So among this, what could be the possible reason that some of the iron reducing bacteria can reduce the uranium? Option A, iron and uranium have similar redox potential. Okay. Any other answer? Any other answer? Option A. Okay. So we will see the solution. Yes, it is correct. Option A. Because what is reduction potential first? So reduction potential is nothing but it tells how a substance has the tendency to gain electrons. Lower reduction potential means they are highly likely reduced. That is, they can gain easily the electrons. And if you see the answer, the uranium, if uranium and iron have a similar reduction potential under the right condition, it can act as an electron donor to the uranium. Thus, it helps in reducing this uranium. And if you see the iron and uranium, they are close together. When they are close together, it is mostly likely favorable reaction to reduce the uranium. Thus, some iron reducing bacteria can reduce the uranium since they have almost similar redox potential. They are under same umbrella. So, if they since the uranium and the iron are same redox potential, the ions will now act as an electron donor, which helps in reducing this uranium. And if you see, if they have similar mass, so this is not going to be directly related to the redox reaction, right? So this is not the correct option. And if you see the structure of atoms is not primary driver for the redox reaction. And if you see the charges, while the oxidation state of iron and uranium are relevant, similar charge does not explain the ability of this iron reducing bacteria to reduce the uranium. Whereas if we have similar size, also as we have explained that mass is not directly determining the reduction potential, same applies to the size. So if two substance, iron and uranium, have the same similar reduction potential, they have the chance to reduce the uranium if they have given the certain circumstance. So the next question is acid mine drainage is the product of option A geochemical reactions, option B methanogenesis, option C chemolithotrophy, option D chemoorganotrophy, option E fermentation. So which is the reason and or what are the product of this acid mine drainage? Acid mine drainage is the product of, so what is the answer? Acid mine drainage is the product of geochemical reaction. Option B methanogenesis. Option C chemolithotrophy. Option D chemoorganotrophy. Option E fermentation. Option C chemolithotrophy. Okay. Any other answer? Option C. Okay. We will see the answer. Yes, it is correct. Chemolithotrophy. And what it is? Chemolithotrophy is a process by which organism will obtain their energy from inorganic compounds like sulfides and ferrous ion, and they use them to synthesize the organic compound. When we try to correlate with acid mine drainage, what happens? Certain bacteria and archaea, they will utilize these inorganic compounds as their energy source, thus leading to oxidation of these pyrite molecules. They release acid water laden with heavy metals. So this oxidation process generates high amount of sulfuric acid, thus contributing to the acidic nature of this AMD. So this is the answer, and the last MCQ for the session is after the Exxon Valdez oil spill in 1989, the fertilizers were added to the affected shoreline. This was done as a part of option A biostimulation, option B bioaugmentation, option C composting, option D landforming. So which is the part of the above incident? Option A, okay. Any other answer? I will repeat the question. After the Exxon Valdez oil spill in 1989, the fertilizers were sprayed over the affected shoreline. This was done as a part of option A. Okay, we will see the answer. Yes, it is correct. Of biostimulation. And what do we mean by biostimulation? We will be giving some nutrients to the affected area to stimulate the microorganism. Right. So it is nothing but promoting the growth of indigenous microorganism to undertake the bioremediation. So in this case, we have added the fertilizers to the shoreline. Why? To accelerate the natural biodegradation of the spilled oil by stimulating the growth of indigenous oil degrading bacteria. So that's why the biostimulation is the correct answer. So these are all the MCQs that I wanted to discuss in the last interaction session. So now we will quickly go through what are all the topics that we have covered so far in the past 12 weeks. And if you see the overall coursework in the first session, it will be like we will spend another 5 minutes so that I will give a glimpse on the environmental biotechnology course, where in the first week we have seen the basics of biotechnology, and we have seen the evolution of this environmental biotechnology. Then we have seen the basic terminologies of ecosystem, ecology, and we have seen several biotic and abiotic factors. And also we have seen what is species, population, community, the hierarchy level in the ecosystem. Then we have discussed what are all the components of the ecosystem, and we have seen the difference between food chain, food web, etc. So these are all the important concepts that we have discussed in the week one lecture. And in the week two, especially we have seen the microbial ecology concept where the microbial species were discussed, followed by species diversity, species abundance, and concept of guild, and what are the microorganism, why microorganism play an important role in the environment, and what are all the micro environment like biotic and abiotic factor. And also we have discussed about the biogeochemical cycle and how the interaction between the microbial ecology and the environmental biotechnology. And also we have seen some fundamental prerequisites of the microbial ecology concept and how it can be tackled. And we have also seen the advancement in the microbial ecology. And in third week, we have seen some of the services of microbial ecology, and we have seen some principles of good management. We have also studied about the mathematical modeling, and moreover, we have discussed uh, the five prerequisites. One is aim for big target. Then we have discussed why there should be development of more powerful tools. Then follow electrons and retain slow growing biomass. And last principle is integrate. So these are all the five major topics that we have covered in the week three. And in week four, we have seen omics related approaches, some goals of microbiology, and growth of microorganism relevant to environmental biotechnology. We have seen various energy sources and carbon sources, and we have also seen groups of microorganism where we have seen prokaryotes, eukaryotes like fungi, protozoa, algae. Then we have also seen the elemental composition of biomass, where we have discussed the growth kinetics, growth media, storage compounds, and classification of microorganism based on their growth, carbon growth. Then we have seen the growth, proliferation, differentiation, and how cells is transformed into specialized cells. So these are all the some key factors that we have seen in week four. And week five, we have discussed the methods in the microbial ecology, where we have studied culture dependent and independent analysis, where we have seen PCR based techniques, microarray, and environmental genomics. And in week 6, we have studied the biodegradation concept and what are the principle of this biodegradation, biodetoxification, etc. And we have seen a common biotransformation mechanism and the effect of organic contaminant structure on the biodegradability. And we have seen various PCR based methods also. And we have seen microscopic techniques and fish fluorescito in situ hybridization technique too. And in week 7, what we have seen the bioremediation concept, where we have seen different types of bioremediation and different concepts like bio augmentation, stimulation, in situ bioremediation, engineered bioremediation, etc. Where in week 8, we have studied the heavy metal problem, why it needs to be remediated, and what are all the different microbial interactions like bio mineralization, bio reduction, bio leaching. These are all the important concepts that has been covered in the metal remediation. And we have seen biohydrometallurgy, their principles, and we have also seen the leaching, their mechanism and methods like HEP and dump bioleaching, and where we have also studied the in situ and bio-based biohydrometallurgy. Followed by we have seen the advantage and limitations and applications. And in week 9, we have seen what are all the different mode of degradation, especially in the aliphatic and aromatic compound. And we have also seen some emerging pollutant degradation and how these microorganisms have interacted with this emerging pollutant like plastics and antibiotics. And week 10, we have seen especially the microbial enhanced phosphorus nitrogen removal, where we have seen the biological way of treatment. And in week 11, we have focused on microbial enhanced oil recovery. And also we have seen carbon capture and sequestration techniques and how it converts these cells into biomass. And we have also seen the MFC, microbial fuel cells too. In week 12, we have seen different case studies. The first case study that we have seen is aquifer chromium bioreduction at Anford 100H using polylactate stimulation. And we have seen the uranium bioremediation through bioreduction that has been occurred in Riffleside, Colorado. And case study 3, acid mine drainage at Malenjukod copper project. And we have also seen the bioremediation of petroleum oil spill, which is deep water oil rhizome. So these are all the case studies that has been discussed in the environmental biotechnology course. So this is the short overview of what are all the elements that are present in the environmental biotechnology course. And you can go through once in order to prepare for your final exams. So if you see the exam date, I hope it has been announced, and I think it is November 1, and you will also get mail regarding this. And if you see the evaluation, first what they will do is they will out of 12 assignments, as you know it is a 12-week course, so in every week you have done your assignments. I hope you have attended all the assignments, but among the 12 assignments you will be uh, taking the best scores of the 8 assignments, and it will be given as 25 percentage coverage. And the final exam will be 100, and from these 100 they will be taking 75 percentage of the mark. So for final evaluation, the average of assignment score and the exam score will be taken for consideration. So this is the exam evaluation pattern. And don't forget to take the all ticket, and this all ticket will be sent via the email ID that you have provided for the registration of this environmental biotechnology course. And also don't forget to take any ID card, government authorized ID card. So you will be getting know your final allotted exam center that will be mentioned in your all ticket itself. You just check your email frequently, And I hope you all know that this environmental biotechnology course is a computer based exams. So there will be maximum MCQ based questions will be there in the environmental biotechnology course. And my advice is to you often visit the announcement tab where you will be getting all the relevant informations regarding the environmental biotechnology. So this is all about the environmental biotechnology course. And if you have any doubts regarding the environmental biotechnology course, you are free to email me or you can put your queries in the discussion forum. And if you have any queries regarding the exam schedule or any other common queries, you can always contact the NPTEL team. So they will try to resolve the issues. So this is the last session for the course Environmental Biotechnology from my side. So thank you. Thank you all for patiently attending all these 12 weeks. And I hope you have attained some knowledge on this course. And as you know, this is an interdisciplinary course. So if you are any branch, it will be useful since we have covered vast applications. So this is all from my side. So if you have any queries, you can mail me. So you just prepare for your final exam and don't forget the hall ticket and ID card and try to reach the venue at least 1 hour before so that it will be very useful for you to attend the exam in a very relaxed manner. So that's all from my side. Thank you all. Thank you, Ashwini, Mridula, and Tuhin for patiently attending the lecture. And I hope you will attend the final exam and get a good grade. Thank you all. Now you can leave the meeting. Thank you, Ashwini. Thank you, Tuhin. Thank you, Mridula.